Thank you, Professor Gooding, for the nice scientific talk. And we open up this session for the Q&A section. Regarding the scientific talk, we have one question that I would like to bring to you. There is a question regarding the technique. Are there any techniques to separate RNA without using magnetic field?
I think people have done that for capture of surfaces, where you you can flow the sample across the surface, but it's quite challenging. But yeah, I think there are ways of doing it where we use surfaces modified with sequences of DNA that are complementary to the RNA you want to extract. But in our case, we feel that doesn't necessarily get us as low a concentration as we really wanted to, in a as quicker a response time as we wanted. I should say that we also do circulating tumor DNA using this approach, where there's also a issue of microRNAs are stable in the blood, but they're in the exosomes, but circulating tumor DNA is not stable in the blood. So it's the time that the rapid response that you get with the magnetic nanoparticles is particularly important. That's a good question.
We have the next question around the scientific talk, and that's apart from the use of nanoparticles, how are other nanomaterial systems being employed in sensing? For example, 2D materials or heterostructures?
Yeah, so that's also a great question. So a lot of 2D materials are being used in sensing, either for improved performance, such as in gas sensing, where you can get improved responses to the gases, but also in optical sensing. A lot of these 2D materials have really interesting optical properties. When you start talking about heterostructures, I think some of the mesoporous materials are also got excellent optical properties. We worked for a long time on porous silicon. So actually, one of our editors, Michael Saylor, is the pioneer of that field, where you can get very precise optical properties. I mean, I think that materials are really, and nanomaterials have really advanced and changed sensing dramatically. And the only the key thing is we've got to use each of those new materials that gives us a capability we didn't have with other materials. And I think there's a lot of nanomaterials that can do that. I mean, we couldn't imagine for a long time having sensors that worked inside cells. And now we have a whole myriad of sensors that work inside cells, because these nanomaterials allow us to go in, send very small sensors inside cells, but still give us information back out through, say, their optical properties. So I think nanomaterial has really changed sensing enormously.
Okay, thank you, Professor Gooding. There is a next question. The work on dark field microscopy, can we combine with electrochemistry to make it quantitative in a sense?
Yeah, that's a great question. Well, I don't think you need to combine it with electrochemistry to make it quantitative, because we think it can be quantitative as it is by counting all the individual particles. But I feel electrochemistry could give the dark field microscopy many great advantages, because you could use the electric fields generated with the potential to control what interacts with the surface and what doesn't. I also think there's very interesting plasmonic properties that change when you apply electric fields to nanoparticles. So it's quite an unexplored area. So I think it's a great question.
So there is one question around the dark field microscopic sensing technique. It's like, do you have ideas of generating a reference-free system to simplify the mapping of the particles before and after the interactions?
Yeah, no, we haven't thought about that. That's also a really good idea. At this stage, we feel that our nanoparticle synthesis, as good as it is not quite good enough to compensate for variance between nanoparticles that are on the surface. But that will be solved. One of the great things about this approach is, because it's single molecule counting, we don't need calibration, which we like a lot. Reference-free is a really good idea. I don't know how much. I mean, I think it really comes down to the magnitude of the signal change. If you can construct assays where the signal change is bigger than the variance between the nanoparticles, then you don't need then you don't need to do the pre-mapping. So it's a great question.
Thank you, Professor Gooding. There is one more question regarding the scientific talk, and it's on the preference in fluorescent versus enzymatic reactions, like colorimetric or luminescence, in the sensing techniques. So this is the question.
Yeah, I've got no preference. I don't necessarily think their fluorescence versus enzymatic is necessarily equivalent. I think that fluorescence is because you've got a dark background, so a luminescent approach is generally stronger if you want to look at really low concentrations of analyte. But colorimetry is also fantastic if you're looking, especially at higher concentrations of analyte. You can it's very simple instrumentation, or you can even use your naked eye. So I really think it comes down to the purpose. I will always say that for most things. If I would use the simplest technique possible to allow me to answer the question I want to answer, or give me the information I need.
Okay. Thank you, Professor Gooding. Now I would like to open up the questions that are journal specific and where our panelists could also give their insights. So one question that I have come up regarding the journal is that what is the scope in ACS sensor in relation to battery, energy, and material research?
Yeah, that's a tough question, but I like it a lot. Maybe one of our EAB members might want to have a first stab at this question. I've talked a lot already. If not, I'll do the talking.
I think either Parmeshwar or you should talk about this.
So Professor Iyer, you are on mute. Please unmute yourself. Thank you.
Can you repeat the question once again? About the battery?
Yeah. So the question is around the scope of ACS sensor over the research articles regarding the battery, material research, and other techniques.
Still, I'm not very clear with the question. If I'm right, are they comparing the?
Yeah. So they are trying to. So they are trying to ask what is the scope of ACS sensor in terms of talking about the research that are generally done in battery areas, energy, and other material research specific areas.
Maybe Justin, you can go.
Yeah, I'll talk about. So talking about batteries, I think the key thing here is that where can I imagine battery research becomes relevant to sensors? And I think battery research is relevant to sensing in the case of wearable sensors, because the batteries are a really big issue. But we're not an energy journal, we're a sensing journal. And so I can imagine that if you're developing a battery that is really portable and flexible and low weight, but high energy storage, then and you've integrated in with a sensing device, I think then we'd be interested, because that's going to advance the wearable sensor field enormously. But if it's just batteries, or it's not going to give us new capability for the sensing, I think it probably doesn't fit into the scope of the journal. And I think I could give the same answer for the materials question. As I said, nanomaterials have revolutionized sensing. But if the paper is about the material, then I would suggest that maybe it should go to a materials journal. But if the paper is about where the material gives a new capability in sensing or solves an established problem in sensing, then I think that's what we're interested in. So if you can tell us a story about how this new material is really good for sensing and gives us a capability or a performance we didn't have before, or solves a problem we've always had, is in the same way that I talked about with the magnetic nanoparticles, gold-coated magnetic nanoparticles solving a problem of response time, then I think we love those papers.
Yeah. Thank you, Professor Gooding. So Professor Iyer, would you like to add something?
No, I think, it was very nicely covered by Professor Justin. The importance of this battery. And, for the wearable devices, I think, it's a very appropriate thing that people are looking as the sensors field moves forward. From normal sensors, as we move on to wearable and implantable sensors, having, good energy storage devices is very important. And so having good materials that can do that, I think it's very important. But as such, the focus should not be on the battery or on the new material, but on the sensing performance. So I think that is the most appropriate thing that this journal is looking for, and not just the material, new material, or the energy storage capacity.
Thank you, Professor Iyer. So moving on to the next question regarding the review articles. As review articles are mostly by invitation, what is the right procedure if an early career researcher is interested in contributing a review article for ACS Sensor?
Yeah, that's from Vishal. That's a really good question. So all review articles I approve whether they can be submitted or not. And I'll often consult with the associate editor if I'm not sure about a given field. But what we're looking for in review articles is we're looking for that they're critical. They're not just a survey of the literature, but they're essentially taking the papers in a literature and from them distilling new knowledge, telling us something that we didn't know. So they're not so different from a primary research article, except that the data comes from other papers. So that's what we're looking for. And so we feel that anybody at any career stage can write such a paper, though, of course, the greater your knowledge and depth of knowledge in a field, the better that paper can be. So they're often for senior people. But we encourage early career researchers to submit proposals. So if you want to publish a review article or a perspective article, you write to the journal, the EIC at ACS Sensors or at acs.org, and then that will come to me, and then I might say yes, I might say no, or I might say I'm interested, but I would like to see more of this, or have you thought about that? And then so you would refine that proposal if that's what I said, and if we give you the go ahead, then you submit it. Of course, that doesn't guarantee that it will be accepted, but generally, if we've had a good conversation about the article, or I've said, yeah, I think it sounds good, that means I'll also send it out for review. The most of the review articles that don't go out for review are the ones which people just submitted without consulting with us first. But I really want to emphasize that writing good review articles is a great way to build your career and build your profile. And we're really keen to support early and middle career researchers as well as senior academics. So if you've got a great idea, a review article that hasn't been published before, write to us and ask whether we'd be interested.
Yeah.
I just want to add at this point that, again, what Justin said, make sure it is not a compendium of the literature, and that's not your review, but it is bringing something to the table which just by reading 3 articles you don't get. Maybe that's the only problem that the early researchers should concentrate on, if possible.
Yes, I do absolutely agree with that. It's a very, very good advice.
So, Professor Iyer, you would like to add anything?
No, I think it was a good explanation.
Yeah. Thank you. So, moving on to the next question, we have what advice do you have for your authors looking to submit to ACS Sensors, and how does it distinguishes from the analytical journals?
Does either of my EAB members, do either of the ACS EAB members want to try and answer this first, or shall I answer it first?
I can try, but actually Justin already answered this little bit during his talk, if you were listening. He said that they collaborated with analytical chemistry, and more industry-related application which has already reached out has been done in analytical chemistry, but new concepts and something that you can perhaps establish, etc., are done in a sensors field. So yes, you can either go in either places, and they said that he just mentioned, it doesn't have to be just in a particular field, but it could be application also. So I think that you can try both journals in this case when you have such a thing. But if you have a basic concept which you are trying to establish, or you're going to try to establish, for example, sensor in a applied system, then ACS Sensors may be the way to go.
Yeah, I would absolutely agree with that. So, Analytical Chemistry as a journal is a great journal, and it's a great legacy journal of the American Chemical Society. So I would say, first off, if you are going to submit your sensing paper, I would recommend you send it to ACS Sciences or Analytical Chemistry, both great places. Why would you submit it to one versus the other? And I think that comes down to, a lot of it just comes down to the audience you want. So Analytical Chemistry publishes great science that puts forward new analytical concepts. So there's a lot of great sensing papers in Analytical Chemistry, and I'm very jealous that some of them weren't submitted to ACS Sciences. But at the same time, and your new sensing concept may be a broader analytical idea. For example, we thought when we first came up with the idea for the dispersible electrodes, that was a broader idea than just sensing. But if you think that you want the sensing people to see your work, and your audience, you want to be sensing people, or you think that the issue that you're dealing with is much more specific for sensing than all analytical chemistry, then we'd recommend submitting to us. So there's very few papers that are submitted in analytical, so it's very few papers submitted to our journal that couldn't be submitted to Analytical Chemistry. There are a few, but there are a lot of papers that you could submit to Analytical Chemistry that you could not submit to ACS Sciences. And one of the things we're trying to avoid, is that the authors try and turn us into a copy of Analytical Chemistry, because that's our parent journal, and we don't want to harm that as well. So it really, for me, it comes down to your audience.
Yeah.
And I think.
Thank you, Professor Gooding and Professor Anand, for sharing your thoughts. And I think, Professor Gooding, I would request if we can take two or three questions.
Yeah, I think Professor Iyer might have had something to add to that last point. I wasn't sure.
No, I think, your lecture slide, as well as the description given by Professor Ruchi and you, I think, they were very good. I don't have to add anything in this.
Okay, sorry.
So, Professor Gooding, I hope you are comfortable with 2-3 more questions.
Oh, yes, definitely.
And our EAB members too. Please confirm.
Yeah, sure.
Yeah, thank you. So there's one question around the expectation from research to be submitted in the letter sections in the ACS Sensors. So please give your thoughts around that.
Yeah, so generally letters are shorter bodies of work. There might be a specific concept that doesn't require a lot of space to answer or to address, and so then you'd go for a letter. We don't treat letters very differently to full articles. We treat them exactly the same. They go through the process as quickly as we can get them through. And so I think it really comes down to just the length of the body of work.
Yeah. So in addition to Professor Gooding's thought, anything from our EAB members?
Well, I only thing I can say is it is a personal decision. What do you think will become as a letter or a full article? And if you have a lot of work, if you have a proof of concept that you just started and you want to put it in a letter quickly, that's a good thing to do. But if you want to extensively put the research, you can make a article out of it. And it is something that you have to decide at any point where your research and how much of a research you are trying to put forth at a particular point. You may want to put it out quickly because you started in a field and you're excited to tell the world very quickly and more work is coming. You can put it in a letter. So, I mean, it's a personal decision, that's why.
Yeah.
Yes, I agree.
Yeah, I agree. I also agree, actually. Unless it is really urgent, and if you are able to put it in a short, convincing manner, I think you should go for a letter. Otherwise, if you think that if you need more, explanation and more description, that will explain your entire result, probably it's not good idea to go for letters. In that case, you need to go for a full paper rather.
Yeah, that's right. And I would say that the secret of writing good research papers, one of the secrets is to write the shortest article you can write that explains the concept and convinces the audience. The least, we don't like papers too long. So if you think you can condense it down to a letter, I think I prefer to read that anyway.
It's actually more difficult to write short and longer, many times.
Very much more difficult.
So your paper will be actually more, eloquent, I would say, if you're talking. I mean,
yes.
Very nicely written. Tight.
Thank you very much. So we'll take the last question, and it's around the batteries and energy storage devices that we had in the earlier discussions. So it's around batteries and energy storage devices need a lot of sensors to analyze their state of charging and for the detecting of the degradation pathways. Would ACS sensors be interested in such concept?
Yeah, generally I would say it's unlikely to be within the scope of the journal, because if you go back to what I said about the scope of the journal, it's detecting biological, chemical, species or process. It's an analytical chemistry journal related to sensing devices. So the sort of information that you get there, which you're talking about, like the degradation of batteries or catalysts, that's super important information. But the focus of the study is in fact the battery, and understanding what the battery is doing. It's not, the focus of the study is not the sensor. The sensor allows you to get that information, but it's not the focus of the study. So think about the scope of the journal in terms of, if I want to detect a species, a molecular species or a biological species, then it should fit well within the journal. But if the focus of the study is to understand batteries, not understand sensors, then it wouldn't fit within the scope of the journal. So it's all about, think about it in terms of the core focus of the paper. That will tell you whether it's a sensing paper or an operando method for understanding batteries. I hope that explains it for the questioner.
Yeah. Thank you, Professor Gooding. Anything that Professor Anand would like to add, and Professor Iyer too?
I don't want to add, but before you end, there is one question that I'm looking at the question answer. So if you, when this is over, I would actually put forth that question because I think it's relevant.
Sure, Professor.
Thank you. I don't have any other questions or answers for anything else.
So I'll ask that question actually, and then maybe all of us can answer it. The question is, what are the key issues regarding editorial acceptance or rejection in ACS sensors, and are there any areas that ACS sensor is planning to focus on? So I'm asking you the question, although it's in a question answer session. So I thought maybe you should answer it.
I think it's a great question. Shall I answer it? Is it for me or for yourself?
No, it is not. First you answer it. We can always talk about it.
So I think that when we don't have any particular area that we want to focus on, I would say that as long as it's a chemical or detecting a chemical or biological species, then we would like to see that. There are areas that I think are really important that are underrepresented in the sensing field, though they're published in a lot of other places. For example, protein engineering. Protein engineering is a way forward for sensors. We've done an awful lot of work on the electronics. We've done an awful lot of work on the surface chemistry. We've used biological molecules to give ourselves selectivity. So it's time that we start tuning the biology to give us better sensing performance. That's one area I'd like to see more work done. And we specifically have an editor for protein engineering because that's an area that I want to see grow. That's Martin Merks in Netherlands. Other area I'd like to see more organoid work. So I like biology, of course. I want to see work. I think that 3D cell cultures and mimics of organs to understand how organs respond, how our body may respond to drugs, or how to design better drugs. I think that's an area I'd like to see more of. I think for biologists, they don't appreciate how much nicer it can be to publish in chemistry journals sometimes than biology journals. We ask for less of you sometimes. Other areas I'd like to see more work. I, as you've seen from my own talk, I really like the single molecule field, but how it can be used for analytical chemistry, not for identifying single molecules, but how we can use it for analysis. I think that's a really important emerging area. I think the other area that I'd like to see more of is more integration of devices. So we do a lot of papers where we show we can detect something in the lab, but taking it that step further to integrating it with the electronics, such as you see in wearable devices. This is a very interesting and important area. I would like to see the issue of selectivity in gas sensors being better addressed. I think there's a lot of great gas sensors around, but gas sensing is so difficult, and how do you make them more selective? I think that's a really important issue as well. So there's lots of areas but I think that really sensing as a whole is such a fascinating time. Obviously, this terrible time we're in with this pandemic really emphasizes the importance of diagnostic devices. So obviously we would like to see technologies that can detect viruses in very quick time, because then we would stop the spread without requiring amplification. That would be a great thing to develop. So yeah, I think there's a lot of great areas in sensing. What we're seeing is that optics is taking over from electrochemistry. The earlier days of sensing was dominated by electrochemistry. Now we're dominated by optics.
Yeah.
Anybody Professor Anand, do you have
Professor
any?
Anand, yeah.
No, I asked the question because I wanted you to answer it for the audience here. So, I mean, you have given a much broader perspective than I would have given.
I realise that, but I realise I didn't answer all your question. I didn't ask the editorial criteria aspect.
So only,
which of course,
because it was a long question.
Yeah, I think in terms of editorial, I mean, I think that we love publishing papers that are new ideas we haven't seen before. So if you think your work is novel, like it's a new idea, a new concept, or you think you've taken an existing sensor and used it in a new way to give new information, they're the papers we love. They will always be looked upon favorably. And in fact, I've talked to you a lot today about the criteria we have in those 10 tips. But one criteria I didn't mention that wins over all other criteria is novelty. If it's a super new idea and you've never put it in a complex sample, but it's really interesting, we still want to publish it.
And like was mentioned before, which I want to stress is that if you make a good sensor system of whatever kind, and you can show its applicability in the right range, not in a range where it is not supposed to be, you have to harp on the point which is on its application in the right sense. And you have to have the right backing and supporting experiments which say that, okay, this sensor is really useful here, and even if it is not perfect, this is what it brings to the table. Which I
agree that it doesn't have to be perfect. It just has to be able to do the job, or show what we need to do to allow it to do the job.
So thank you, Professor Anand, for taking up the question, and Professor Gooding for answering the question. Thank you very much. So once again, on behalf of the audience, thank you, Professor Gooding, for such an insightful talk and for sharing your thoughts and vision as an EIC of journal ACS Census. We hope our EAB members, Professor Anand and Professor Iyer, enjoyed the Q&A session. And
thank you.
Just to update you, we had around 100 participants in this particular session, and we'll be sending the certificates to all the attendees via email. Finally, we would like to thank the audience for their time and the wonderful participation. And we'll be back again with another session in the ACS Science Talks session. So till then, stay healthy and stay safe. Thank you so much, Professor Gooding, Professor Anand, Professor Iyer, for taking your time and joining with us in today's ACS Science Talk session. Thank you very much.